Today I am presenting one of the technology from our lab like LB-PON as a prophylactic agent against organophosphate nerve agent poisoning like Prativisha. So the problem organophosphate nerve agent organophosphates nerve agents are exploited as weapon of mass destructions and in terrorist attacks and chemical warfare. So moreover, slides are not. Okay. Sorry. So organophosphate nerve agents are exploited as a weapon of mass destruction in terrorist attacks and chemical warfare. So moreover, a constant threat of intentional OPNA exposure to civilians and military's personnel exists in the current world scenario. So existing treatments of both prophylactic and post-exposure of organophosphate nerve agents are limited in their effectiveness and often come with wide range of adverse effect. So organophosphate nerve agents are the highly toxic chemicals for human and need of an hours for development of effective and safe antidotes for organophosphate nerve agent poisoning. So there are different classification of nerve agents like G agents, V agents and A agents currently developed at Novichok agents. There are different subtypes also, but we are not going into that now. So the solution and scientific validity towards this, constantly research efforts are focusing on the development of protein-based scavengers to serve as antidote to. Okay. So towards this, the US Army first initiated with the development of human butyrylcholine is being developed as a prophylactic agent for organophosphate nerve agent poisoning and approved under trade name of Protexia. But in 2009, they completed the phase 1 trial and approved for the emergency use. But the major problem is with recombinant human BUCHE can bind only OP and OP organophosphate nerve agent as a 1:1 like stoichiometric ratio. So it needs more dose like 400 to 700 mg per human dose for giving an optimal protection against nerve agent. To address this issue, the catalytic bioscavenger are being developed and they are considered as the next generation bioscavengers antidote agent. Towards this, our lab is developing a recombinant human peroxonase from since 2008. So recently I joined this program, but recombinant human PON1 suffering with serious limitations like low nerve agent hydrolyzing activity and poor pharmacokinetic activity. We thought for recombinant human PON1 is like our human protein first synthesized in the liver, and when it associated with high density lipoprotein, then it will circulate in our body. So recombinant human PON1 can bind more than one molecule of organophosphate nerve agent like bioscavenger, also known as catalytic bioscavenger. So, but the development of catalytic bioscavenger against organophosphate nerve agent poisoning is accepted and validated approach. Towards this, different agencies like US Army and Israel Army working for this from 2001 onwards. So the solution, the solution and scientific validity. So recombinant human PON1 has two problems: poor pharmacokinetic activity as reported in literature. It can stay only 3 hours in human body, so and less nerve agent hydrolyzing activity. Towards this, we use the fusion protein approach to increase the half-life of protein. We use recombinant human PON1 with fuse with non-recombinant human PON1 protein with other body proteins, and we use some mutagenesis approach to increase the OP or organ-phosphate nerve agent utilizing activity. Then we got this molecule and we patented, and we got the trademark also albumin peroxonase. So then we tested this molecule in SD rat animal model. So we used as a control vehicle plus upon exposure of organophosphate. Now currently we are not using any nerve agent in the lab. So these are the very restriction chemical. Only defense can use, like in our country DRDO can handle it and other defense agencies. So in our lab only surrogate is there, parathione derivative is there. So we use that. So upon exposure of peroxone, rat is dead. Then previously idea is like that ki LB-PON can be developed as prophylactic. Prophylactic idea is that if we injected LB-PON like other prophylactic agent in the body, it will circulate in at least time of 1 day, 2 day, and it can move, and the small operations can be done by our forces. So but LB-PON we tried at 2 hours before prophylaxis, and then addition of the toxin peroxone, then mice will be protected. So prophylactic protection by LB-PON is there. We published the paper of this. So limitation of Protexia are overcome by LB-PON. So work done so far for development of LB-PON. Okay, so we engineered the novel recombinant human PON1 variants. So panels of novel variants of recombinant human PON1, more than 30 variants are designed. Panel of variants are produced and characterized one lead candidate are identified like LB-PON. So next, like clone and process development, we are producing this recombinant protein from E. coli. So clone develop and industrial feasible lab scale production process of LB-PON is also developed in our lab. So next step went for characterization of this molecule. LB-PON exhibited enhanced OP hydrolyzing activity in our lab. So in vitro hydrolyzing activity peroxidase. Then LB-PON exhibited enhanced pharmacokinetic properties in SD rats. We reported 25.1 hours pharmacokinetic property in SD rats. Then we checked the LB-PON prevent the development of organophosphate poisoning symptoms and mortality in animal with control with recombinant human BUCHA on other approved drug for prophylactic and post-exposure treatment for organophosphate nerve agent poisoning. So then we don't have this nerve agent in our lab, so we send this sample to France Army, IRBF France. So they check the in vitro nerve agent neutralizing activity in buffer. So we got the results. So all G-type nerve agent hydrolyzing activity of LB-PON. We haven't reported this result anywhere. So POC established. So these are the patent and papers are reported from our lab, and these are the prototypes. So we are sending with our collaborators every time with these vials and all. So current TRL status is like 4, and cost analysis ratio, if we consider it like in our lab scale process protocol, so it is around 9,000 to 10,500 rupees we need for production of 1 milligram of protein. So what is road ahead? So currently we just done with the surrogate study. So we want to test the efficiency determination of LB-PON to prevent nerve agent toxicity in vivo. Currently we are working with DRDO Gwalior for this. So next step will be the dose optimization study with available chemical antidotes. With maybe some synergistic effect will be there. So we want to check that also. So next we obtain the dose, then we want to go for the non-human primates like monkeys. And then after that we will go for safety pharmacology, and simultaneously we will go for production process and GMP level production of LB-PON. Then final data generation and IND filing. Our expectation is like industries from joint research and technology transfer. So market potential, since there is this antidote against organophosphate nerve agent, there is not that much huge market. But since the this is like it can come under emergency situation like COVID-19, like eye opener, you should prepare for next any attacks and other things for our nation's defense point of view. So thank you.